morning, everyone. Today we have Tanmay, who's a postdoc in the Jan Lewis group. Whereas perhaps most of the course has been dealing with single particle analysis, we'll make a little step sideways today, and Tanmay will discuss tomography. And Paula, in her lecture, already mentioned that tomography, subtomogram averaging, could be used for initial models for single particle analysis. But there are many problems in biology where it's not easy to make many copies which are structurally the same. So you could do averaging over projections of copies of the same structural thing. But if there's something that doesn't repeat itself, like an entire cell or something, an organelle or so on, then tomography provides the unique opportunities to study that. But I'm sure Tanmay will discuss that with you.
Thank you very much, Shores. As Shores mentioned, we've had a few lectures on single particle analysis. Today I'm going to tell you about a very exciting application of electron microscopy, which is electron tomography. So in tomography, we take many different images of our sample in different orientations, and then we combine all these images back into 3D to get a representation of a three-dimensional object. So let's assume this is our EM grid. A typical tomography data collection would include taking an image, tilting the stage, taking another image, and so on and so forth. Then we get our tilt series, and then this tilt series is back projected into 3 dimensions to get our 3D structure, which is our tomogram. So here's an example of a tilt series that can be acquired in an electron microscope. And the principle of 3D reconstruction is analogous to single particle analysis. And after we have done our reconstruction, we end up with a volume like this. And this volume, 3D volume, which is the result of our calculation, is the tomogram. This is our aim. So this is a slide from Shores. In single particle analysis, of course, we don't have very good knowledge of the orientations. So the single particles can fall randomly on a grid, and they can be oriented in many different directions, and these orientations are not known previously. In tomography, however, we have a very good prior information on the orientations because we know how we've tilted our grid and acquired the image. So this prior information is very good. So the problem is a bit simpler in tomography. Tomography is associated with the same problems of sample preparation, data collection, and data analysis. And this is the challenge that I have with this lecture, to go through all these steps in one lecture. So hopefully by the end of this lecture, you'll have an overview of what's involved in going from a sample to a final high-resolution structure from your tomography data, if you so require. Okay. This is great. To what can we apply tomography? Well, the answer is, if you can get your electron beam through your sample, you can acquire a tomogram. So tomography allows us to get high-resolution, three-dimensional snapshots of samples that are not easily accessible by single particle analysis. For example, if our sample is arranged in a complex three-dimensional shape, like here, these are polyribosomes, which are in the act of translation, and this is not, with a single cryo-EM image, it's not easy to see in what way the ribosomes in this polyribosome array are arranged. But if we take a tomogram, then the arrangement of this assembly becomes clear. We can look at dynamic in vitro systems, which are not easy to assess by single particle analysis. We can look at cellular processes like snare-mediated membrane fusion, for example. We can look at asymmetric viruses. In this case, our feature of interest, which is the virus capsid, is buried deep within the interior of the virus. And a single particle image, you would image through the entire volume, and you would not, the feature of interest would be blurred. Therefore, we need to acquire a tomogram to slice through the unwanted material to get to the region of interest, which is the virus capsid. So here are some examples. Electron microscopy is a very nice tool because we can, on one hand, get high-resolution information from single particle analysis, but using tomography, we can put this high-resolution information in the physiological context the context of the cell. We can image mammalian cells, bacterial cells, and also cellular components which have been purified. We can even go one step higher. With tomography, we can image tissues and also whole organisms, for example, zebrafish embryo, which is shown here. Okay, so this is great. We can apply it to many different samples. So how can we make a sample which is worthy for tomographic data collection? Well, the answer is, if we are looking at a sample of purified proteins or purified viruses, then we can simply make the sample the same way as one would do for single particle analysis. This is a slide from Tony Crowther's talk, and the sample is simply, you should remember this, the sample is simply applied to an electron microscopy grid, plunged, frozen in liquid ethane, and then imaged at liquid nitrogen temperatures. One difference in tomography is that we often have to add some fiducial markers to our sample, and these can be gold nanoparticles which have just been added at the end, just before plunging, and these usually don't interfere with the sample. If we want to look at whole cells, then we can be lucky, like in bacterial cells. We can simply plunge freeze them, and these bacterial cells are thin enough for the electron beam to get through, and we can simply acquire a tomogram of these cells. If, however, we are unlucky and we are imaging mammalian cells, then we can only image the bits which are thin enough for the beam to get through. So here's an example. This is a mammalian cell which has been infected with Marburg virus and grown on an EM grid, and then plunge frozen like a normal cryo sample. This side is the interior of the cell. It is too thick for electron microscopy imaging, so this is inappropriate for tomographic data collection. However, this virus buds from filopodia-like protrusions, and these protrusions are thin enough, and here we can collect tilt series data and get high-resolution tomograms. If our sample is too thick, all is not lost. We can still cut our thick samples into thin slices and image each of these slices and get three-dimensional images of each slice, and then put them all together in a large montage and get an idea of the entire three-dimensional picture. So this is not cryo, so this is a method in which our sample is embedded in plastic and imaged with tomography. So the method is freezing our thick sample at approximately 2,000 times the atmospheric pressure. In these pressures, the water around the sample does not become crystalline, doesn't make crystalline ice. The sample is preserved in vitreous ice. Then at low temperatures, the solvent in the sample, which is water, is substituted for an organic solvent, followed by low temperature embedding into a resin such as low acryl or epon, and then thawed to room temperature. And at room temperature, the thick block, which is embedded in plastic, can be sliced using a diamond knife, and the thin salami type slices, you can think of it as a salami slice, and these slices are then put on an electron microscopy grid, and then they can be simply imaged like a normal room temperature sample. One thing I would like to mention is that during the procedure, a contrasting agent is often added, has to be added to see the ultrastructure details, and this contrasting agent could be uranyl acetate or something like that. So again, we are not, the density that we see is the density from the stain and not from our biological material. So even though we can see a large amount of details in the ultrastructure, this is not a structural biology technique. As an aside, and for completeness, I would like to mention that because in electron microscopy we don't have a GFP equivalent, we don't have a technique by which we can localize different proteins, we can still do immunolabeling to localize our proteins in these plastic sections. So there is a method by which we can section our samples in cryo temperatures and add a cryoprotectant, thaw it to room temperature, and then add an antibody which recognizes our protein. And this antibody is coupled with a gold nanoparticle, which allows us to localize where our protein is. And then after that, we can add a contrasting agent, followed by embedding and imaging at room temperature. So that's just an aside. If we want to preserve, if we want to image not the stain in our sample, in the thick sample case, then we have to use a method known as semovis, in which the sample is high pressure frozen and then sliced at cryo temperatures using a diamond knife. And these cryo sections, as they come off the knife, they are put immediately on an electron microscopy grid. They are maintained at cryo temperatures, inserted into an electron microscope, and then we can just do standard imaging like you've heard the entire week. So this is a method where you could do structural, high-resolution structural work because the diffracting density is from your sample and not from a contrasting agent. Although I should mention there are artifacts induced by the diamond knife, which is used to cut the cryo sections, and these distortions are very difficult to correct for because they cause expansion and contraction of the sample, and this we cannot recover. Fine. So now we've made a sample. How can we get the data from this sample? So you will remember from Laurie's talk the principle of low-dose acquisition. We are going to, for tomography data collection, we use the same principle. So this is an electron microscopy grid. This is a carbon support film. These round things are holes full of vitreous ice and our sample of interest. This black line is let's assume this black line is our tilt axis. And so to collect a tilt series data, at this point, we would have to acquire many different additional images at a region which is slightly further away from our sample. For single particle analysis, this additional data was in the form of focus images, which would allow us to apply the correct defocus on the microscope. So this is an example of this additional image, a series of an additional image, and this is the region of interest that we want to target. So these additional images are important because they allow us to track our sample during tilt series data acquisition, allowing us to keep the feature of interest in the field of view. So a typical tilt series data collection scheme would go like this. You would take a tracking image, tilt your stage, take another tracking image, and then cross-correlate the two tracking images to bring the feature of interest back into the field of view. Then take a focus image to apply the correct defocus on the microscope, and then take the next record image. This process is iterated till the entire tilt series is collected. Applying the correct defocus during data collection is important because the tilt axis of the grid does not necessarily pass through the region of interest, the region that we are imaging at that particular moment. So it's important to focus, apply the correct defocus at regular intervals during data collection. Okay. So this is an example of a tilt series from plus 30 degrees to minus 30 degrees. So what you will see is that at high tilts, the image becomes fuzzy. This is because if you're imaging an electron microscopy grid, and when you tilt it, the effective thickness the beam has to travel through increases, and this leads to increased inelastic scattering of the electrons, leading to lower signal to noise. This, of course, also makes a limit on how far we can tilt our stage. So let's imagine that we were to tilt our grid 90 degrees. Then the electron beam would have to pass through the entire width of the grid, and that's clearly not possible. So practically, tilt series data is acquired from plus 65 to 70 degrees to minus 65 to 70 degrees. So if you remember from Tony Crowther's talk that two-dimensional projections of our three-dimensional object are simply slices. If you take a Fourier transform of these projections, they are simply slices in the three-dimensional Fourier transform of our original object. So you can imagine if you can't tilt the grid beyond a certain point, there will be missing information in Fourier space, and this will lead to artifacts in the resulting reconstruction. To illustrate the kinds of artifacts which are caused by this missing information, I will go back to the two-dimensional case. So this is our original object, and this is the reconstruction if complete, if we could fill in Fourier space completely. And here are images which show the effect of increasing missing information. So in this case, there's missing information in that direction, and the effect is particularly severe. And the features along that direction become smeared out, and this leads to artifacts in the reconstruction. So the important thing to note is there is missing information which we cannot recover because it's just missing. So in Fourier space, this is what the CTF of our data collection would look like, the Fourier transform of a data collection would look like. So let me explain that. So each slice represents the Fourier transform of one tilt series image. So these slices are equally spaced. This is because when we collected our tilt series, the tilt angle was constant, and this position where there are no slices, this is the point where the missing information is present because we couldn't tilt our grid. There are many different ways you can design a data collection scheme. You can have a linear tilt scheme like this, or you can have a more complex tilt scheme where you can upweight the higher tilts, and there's still discussion which one is better, which one is worse. There's a way to circumvent the missing wedge altogether. Jan Löwe in the lab, my boss, has designed the cylindrical specimen holder, which is cylindrical, so you can tilt it much more, and it allows tilting the stage to plus minus 75 degrees, reducing the missing wedge and reducing the artifacts in the final reconstruction. However, of course, for routine applications this is not possible, and routinely tilt series can only be acquired to plus minus 65 degrees, or close to that. One last thing I would like to mention, this is a slide from Chris Russo's talk, that while collecting tomographic data, it's very beneficial to apply energy filtration, because especially at high tilts where there is low signal to noise ratio, the energy filter helps us in removing the inelastically scattered electrons, thereby increasing the signal to noise in our images, and this has a very beneficial effect, especially at high tilts, and allows us to see our sample. Okay, fine. So now we've collected our data in the microscope. How should we make tomograms? So if you remember, in the beginning I said we need to add some fiducial markers to our sample, and these fiducial markers can simply be these dark gold nanoparticles. These gold nanoparticles are spherical in shape usually, and thus makes them easy to track, because at different tilts, projection of a sphere is a circle, and this is the same in all directions, so it's very, so you can determine the center of these circles quite accurately and track each of these gold nanoparticles. So the first thing to make a tomogram is to align our tilt series. So the first step is usually pre-processing to filter out all the X-rays in our images, followed by a rough alignment. And then after this rough alignment, we have to detect features of interest, which is our gold nanoparticles. And then these features should be corresponded with each other in the entire tilt series. And this is usually done in a semi-automatic way, where the user just clicks through the tilt series and says this gold particle in this image corresponds to this gold particle in the next image, and so on and so forth. Based on this tracking, a projection model is made, and based on this model, we can go from the original tilt series to an aligned tilt series. Yes. So now we have our aligned tilt series. All that we need to do is back project this aligned tilt series into 3D to get our tomogram. Of course, as I said, in single particle analysis, the orientations of our images is not known. However, in tomography, we have a very good idea of our orientations of these images because we know how we tilted our stage. So the most common method of tomography reconstruction is known as weighted back projection. And I'm going to explain this by reducing the dimensionality of the problem. Let's assume that our sample is two-dimensional, and it's this box with a circle in it. So we could take projections of our sample in this direction, in this direction, in this direction. So these one-dimensional projections are like us taking tilt series images in our microscope. So this is our tilt series, this is our original sample. So this is what we have in typical tilt series data. So how we would reconstruct our tomogram is to take these one-dimensional projections, simply smear them back into the two-dimensional volume, back in the direction where they came from. And here is an example using 3 different projections. And if you combine many different projections, we get a much better representation of our original object. And this technique is called simple back projection. If, however, we apply an appropriate filtering or weighting function to our one-dimensional projections, that is our original data, we can do the same thing and get a much better representation of our original object, and this is known as weighted back projection. Practically in tomography, we use prior knowledge such as the size of the reconstructed object, and these can minimize the effect of the missing wedge. But as I said before, they cannot compensate for the missing wedge because it's missing information, we can't get it back. So now we've gone from an aligned tilt series to a tomogram. So 3D map, which is our tomogram. This movie shows, is just a movie of sequential Z slices through our density. And you can see that it seems to have worked quite well. Another method to do tomographic reconstruction is SERT. So it's like weighted back projection in real space, but it's based on an iterative procedure where we compare the projections of the density with the original projections, and this way we can improve the reconstruction. We should not apply simple direct Fourier inversion like we do in single particle analysis, because in tomography, as we tilt our grid, there are different parts of the sample which come into the field of view, which should of course not be back projected like you would in single particle analysis. So we should not do a direct Fourier inversion for tomographic reconstructions. So I would like to mention some cautions associated with tomography data. There is currently no good method for determining resolution of single tomograms. One can see high resolution features of the order of 20 to 30 angstroms just with the naked eye by visual inspection of the tomograms. However, we can't do a Fourier shell correlation like we can do in single particle analysis because it's a unique object. So this must always be kept in mind while analyzing any tomography dataset. The other thing we should always keep in mind is that there's limited resolution in Z, in the beam direction. There's very good resolution in XY, and this must be taken into account when making any interpretation with our data. Okay, so now we have our tomogram. What should we do with our tomogram? Well, the answer is that if the question, the biological question that is asked, is answered by the tomography data, then you can simply stop, look at your tomograms and interpret your density. If, however, you want to do structure determination from your tomographic data, then we have to apply the technique of subtomogram averaging. Subtomogram averaging is simply averaging of three-dimensional volumes taken from a tomogram. So let's assume this is our tomogram, and this tomogram has some macromolecule that is present in many copies inside this tomogram. Then what we would do is we would go into our tomogram, extract the multiple copies of the macromolecule that we are interested in, and these extracted volumes are called subtomograms or subvolumes, whatever you prefer. And of course, in these subvolumes, our macromolecule of interest is arranged randomly. And these randomly oriented volumes can be aligned to a reference, and this reference can be from single particle analysis from the PDB, or it could simply be a sum total of all these extracted subtomograms. It's aligned to a reference, and after alignment, we get some aligned subtomograms. These can be averaged, and this whole process can be repeated till we get our final structure, which is hopefully a higher resolution structure than what you can see with your eyes in the tomogram. Okay, so the first step in subtomogram averaging is to extract our particles. So you can be quite lucky in that in your tomogram, your particles are not so many, and they are quite well separated. Like in this case, this is a tomogram of a hepatitis B capsid, which forms these nice icosahedral cages. These are very easily visible in the tomogram, and one can simply go and click them, and this is an easy case. So you can extract your tomograms manually by simply picking on the tomogram. If you're lucky, your subtomograms can be arranged in geometric shape in the tomogram, for example, with a spherical geometry or tubular or helical geometry, and you can exploit these geometries to extract the subtomograms. This offers a big advantage because the geometric arrangement allows us to define very good prior Euler angles, and these prior Euler angles allow us to do a reference-free reconstruction, and they also allow fast alignment and averaging because we know the Euler angles already quite well because of this kind of arrangement. If, however, we are very unlucky that we can't pick our subtomograms manually, and they are not arranged in any geometric way, then we would have to rely on automatic particle picking. This is analogous to the two-dimensional case. Let's assume this is our tomogram, and inside this tomogram there are lots of ribosomes. So to pick these ribosomes, one would take a three-dimensional model of the ribosome from the PDB or from an electron microscopy, EMDB. Then we would cross-correlate that density to the tomogram, and then using some sort of cutoff function, or we would determine the cross-correlation peaks inside this tomogram, and then using the peak assignment, we can go into our tomogram and extract the subvolumes, the subtomograms, which can be used for subtomogram averaging. One thing I would like to warn you about is that any cross-correlation based picking can lead to this Einstein from noise problem. This has been well documented in this course. And if you cannot see your particles in the tomogram, and you are using a reference to pick your subtomograms, this will make your extracted particles look like the model that you've used. So one needs to be very careful about that. In this case, this is a tomogram of a phantom cell with two distinct components, proteasome and thermosome. In this case, it's a very easy problem because it was known what's actually inside these phantom cells. So in this case, it was appropriate to apply template matching to pick the subtomograms. On the right here are cases of microtubules in bacteria. In this case as well, because we can see our feature of interest, if we use automatic particle picking, which is known as template matching, that would be appropriate because we can visually look at our results and see whether it's correct or not. Okay, so now we have our subtomograms. How do we get a structure from these subtomograms? Well, the first thing to do is to, like in single particle analysis, is to estimate the CTF in our subtomograms. So this is a slide from Tony Crowther's talk. And if you recall, because of the CTF, there are reversals in contrast at different frequencies in our data. And it's important to correct for the CTF to allow us to get high resolution information out of these of our tomographic data. So CTF correction is slightly more complicated for tilted data, and the defocus of a specimen depends on the tilt angle. It depends on the distance of the specimen from the tilt axis, and also depends on the position of the specimen in the ice. So let's assume this is a tilted grid. A specimen which is closer to the tilt axis would have a different height than a specimen which is farther away from the tilt axis, and this of course needs to be considered while doing the CTF correction. And the same thing applies for samples which are in different positions in the ice. Yeah, so we have to take that into account while doing CTF correction. In tomography, because we are imaging the same kinds of samples which are in the same way limited by dose, the dose in each single tilt series image is a fraction of the dose we use in single particle analysis. If you remember from Paula's talk, the way to estimate the CTF is to take a Fourier transform of the image and inspect the tone rings which are present in the Fourier transform. And using this, we can estimate the defocus of the microscope when the data was collected. In tomography, from single Fourier transforms of single images, it's very difficult to see these tone rings. So what one could usually do is to average the Fourier transform, average the power spectrum, which is the amplitudes of the Fourier transform from many different tilted images. If we average the power spectrum, we can boost the signal, and then we can have a much better visualization of the tone rings, allowing us to find the defocus of our tilt series. The disadvantage of this method is that we have one value which we would fit to the entire data collection, which may or may not be true, depending on how stable your microscope was during data collection. Another method to estimate the CTF is to collect additional images. For example, so this is an EM grid again. This is the area of interest where we actually want to acquire data. So the option is to acquire extra images on either side with higher dose. In these extra images, because of the higher dose, you have a much better visualization of the tone rings, allowing you to back calculate what the defocus was in your record area. So this is another way to get the defocus of the microscope back for CTF correction. So we've estimated CTF, and we can do some sort of CTF correction. Now we need an initial model. So reference-free reconstructions are possible in some cases, like shown here, but certainly not all cases. In many cases, it is not possible to do this, and a reference model is needed from some other methods. And Paula presented these other methods, and you can use them to get an initial model. I should mention again that if your subtomograms are arranged in a geometric way, like arranged around a sphere or arranged in a tubular manner, then it's much easier to get an initial model because you know some prior angles. Another advantage of using subtomogram averaging for structure determination is that the hand of the structure is determined absolutely, and assuming you didn't do anything wrong, it should be correct. So we have our CTF correction, we have our initial model. Now all we need to do is alignment and averaging. And this was presented by Shores yesterday, and I'm now going to go into a lot of detail. So 3D alignment and averaging is an extension of the two-dimensional case, and we can apply maximum likelihood algorithms like in Reliant, for example, and then we can do 3D classification, 3D structure determination. But I'm not going to go into detail in this. One thing I would like to mention is that tomograms are already three-dimensional representation of our data. So we can go back to our tomograms and look at the final aligned positions of our subtomograms, and this can help us in assessing how good our alignment is doing. So let's assume, so this is a slice through a tomogram of a tubular assembly. So this is a tube, and we've extracted subtomograms centered around these red dots. Then we can do alignment and averaging, and the X, Y, Z positions of the subtomograms move during the alignment procedure, and then we can plot them back onto our tomogram. And this shows a nice helical arrangement, and we can use this to assess how good our alignment is doing. And one should always do this, plot our aligned positions back onto the tomogram to see if it makes sense or not. We can, of course, do the same for Euler angles, and instead of placing a spherical dot, we can place a more complex shape back onto the tomogram and see whether the Euler angles make sense or not. And they should make sense. If they form a lattice, then the adjoining subtomograms should be in the same plane. Okay, so now we have a structure. Can we publish it already? No, because we've done a structure determination from electron microscopy data, we need to validate our structure. And validation will be presented by Richard Henderson tomorrow. So before publishing your subtomogram averaging structure, you need to wait for his lecture. So, okay, so do I have time? Yes, okay. So let me summarize. Electron tomography data can be collected with almost the same microscopic setup like we have for single particle analysis. Electron tomography can be used to get high resolution three-dimensional pictures of our sample, which are not available from any other technique. And we can use subtomogram averaging in the same way as single particle analysis to resolve high resolution structures within tomograms. So subtomogram averaging is a comparatively new field when you compare to single particle analysis. And there are many exciting developments that are happening in tomography as well as single particle, as well as subtomogram averaging. So I'm going to present two studies that were published recently, which I think are quite exciting. And the first of this is a study using a hybrid light microscopy, electron microscopy technique. So the authors wanted to study endocytosis in yeast. This is a dynamic process, which is very difficult to study. If you just put yeast cells on a grid, it's very difficult to catch an endocytosis intermediate because first of all, you wouldn't know where to look for it. And it's a very fast process, so chances of finding it would be very low. So different endocytic proteins were tagged with GFP and M-cherry. And based on the localization of different signals, the point in which the process was could be defined. So what the authors then did was to make plastic sections of the yeast of the yeast cells, put it on an electron microscopy grid. So this is an EM grid with the yeast cells on it. This is a grid bar here, and the thing which helped to apply this method was the fact that the GFP and the M-cherry fluorescence was preserved in these plastic sections. And using this fluorescence, you could focus in on a region of interest, for example, that cell, and for example, that position, which is some sort of endocytic intermediate. And even though you have a low resolution light microscopy picture, you would like to have a high resolution EM picture of the same thing. Some fiducial markers were applied previously to the cryo-EM grid. These were simply fluorescent polystyrene beads, and these fiducials helped to calculate a transform between the light microscopy image and the electron microscopy image, allowing the authors to define the region of interest to high accuracy, so an accuracy within 100 nanometers. And then, once you've defined your position of interest, you can simply go into the electron microscope and acquire a tomogram. And in the tomogram, you can clearly see it's a yeast cell, and this is an endocytic site. And this same thing you can do for many different states and define a dynamic process which is occurring in cells by the use of tomography. I should mention that this is not possible without tomography. If you took single images, you would not see that. You would have to take a tomogram to see the entire density. So I think that's one exciting thing, one exciting application of tomography, which could become very important in the future. Another exciting study that I would like to present is the use of phase plates to collect tomography data. So there are hardware improvements which are, so the electron microscope is constantly being improved, and these hardware improvements benefit not only single particle analysis, but also tomography. So if you remember from Shore's talk, and also Tony's talk, I think, the CTF has a characteristic of a sine wave, and at low frequencies, the intensity of the signal is quite low. We can use a faceplate, in this case a Cernica faceplate, to shift the CTF to have a cosine like curve, and of course then you can image much closer to focus and have high signal in your data. So this is an image collected using that kind of a CTF, and this is image of the same place collected using that kind of a CTF with the faceplate. So the authors here, they focused the beam, so they are not operating in defocus anymore. With any sort of defocus applied, this is all in focus, and in focus you get a lot of contrast and you can see many different details. And then they went and collected tilt series data of this kind of a sample, and you can see the contrast is just simply amazing. And then using this tilt series data, they could reconstruct tomograms, and in these tomograms you can find, you can see a lot of ultrastructure in these bacteria, you can see ribosomes, and you can see what they originally wanted to see was the infecting phage in these cyanobacteria. So they could identify intermediates, phage intermediates which were about to enter the cell, had entered the cell, and they could even do subtomogram averaging to define different states of the phage during the infection cycle. So let me end with the future outlook, which is quite bright for electron tomography, I think. I think there are going to be many improvements in hardware and the microscope, which are going to lead to improved tomograms, which will allow us to see high resolution features in our data. Hopefully, sometime in the near future, there will be atomic resolution structures available from subtomogram averaging, and this would provide high resolution snapshots of macromolecules in the physiological context. And as I presented in this case study, I think hybrid methods will provide the time dimension and allow us to get biological insight far and beyond that can be provided by electron tomography. Thank you.
Thank you very much, Tanmay. Question? Yes, Lou?
Would faceplates would be useful for single vertical analysis?
I don't think so, because the faceplates are not really a high resolution technique, and you can get high resolution without a faceplate. And let's just say they haven't been proven to give a high resolution structure yet. So until someone solves the high resolution structure with a faceplate, I think it would be better to stick to normal imaging. I think the highest resolution structure which has been solved with the faceplate is around 8 angstroms. So I think the normal way for single particle is better.
When you went from your tilt series to tomogram,
Yeah.
when you looked at the fiducial markers, they were well defined in the tilt series.
Yes.
Completely smeared out in the tomogram.
Yeah, that's because we don't have good as resolution in the Z direction. That's causing them to smear out. I mean, this is a single image, so this is just a projection, and then this is after back projection. We have some missing information in that direction that's causes them to smear out. If we had a perfect tomogram with no missing information, let me go back, sorry. There. So let's say that's a gold particle. If we had a perfect tomogram, we would get our gold particle back. But if we have, this is a typical data acquisition, plus minus 60 degrees, then they would smear out.
More questions? If not, thanks again, Tanmay.
Thank you.